So friends, now we will discuss about different types of plant tissue cultures. We can broadly categorize the different types into two broad categories, that is how you maintain the culture which you are growing, and we can also categorize plant tissue culture on the basis of the plant part or the explant you are using. Now on the basis of the maintenance, we have broadly two types of plant tissue culture, that is callus culture and suspension culture. Whereas on the basis of the explant or the plant part used, we have root cultures, we have shoot cultures, we have embryo cultures, we have anther cultures, we have endosperm cultures, and so on and so forth, depending upon the type of the explant you are using. So now we will discuss in detail different types of plant tissue culture, starting with the callus culture. Now before we further dwell into how the callus culture is different from suspension culture, first we have to define what is callus. So as you can see in the picture, it is just a mass of the cells or unorganized aggregation of the cells. Now callus can also be defined as an undifferentiated mass of the parenchymatous cells without any structural order, as you can clearly see in the picture. This is just a group of cells which have capability to divide. Now there are different types of callus cultures depending upon the stage of the culturing. We have primary callus and we have secondary callus. Now primary callus are those which are the initial cultures developed when the initial explant is subjected onto the culture media and we get an undifferentiated mass of the cell. We call it as a primary callus. Now when this primary callus is further cut into smaller parts and is placed onto the fresh media, that is subculturing, and by subculturing whatever callus we obtain, we call it as a secondary callus. Now we can always maintain callus cultures for longer durations of ranging up to years, but it has been observed that long subculturing of these primary calluses or secondary calluses or calli leads to certain genetic modifications in the daughter generations, and such genetic variations we call it as soma clonal variations. Many times such mutations or genetic changes are quite beneficial and we select the mutants having beneficial genetic variation or soma clonal variation, and we can use them to develop new varieties. We can also classify callus on the basis of their physical appearance. They can be compact, they can be friable, and when they are compact, it has been observed they are quite darker in color or sometimes they are light in color, and same happens for the friable callus. It has also been observed that such calluses can be wet or dry, and by and large most of the dry calluses are more friable in comparison to the wet, which are more compact in nature. Now if we look into the development of callus, we will see that whenever sterilized explant is placed onto the media, and obviously we studied under the component of the media that it contains various growth regulators. So depending upon the outcome we require, we modulate the type of the growth regulator which we want to incorporate into the culture media. But to initiate the division of the callus, we must have auxins in our culture media, which will lead to the initiation of the callus development. It has also been observed, as we discussed, I will again go back to the preparation of the explant, wherein we sterilate, we cut that explant into small pieces and then we place it onto the media. It has also been observed that the development or the initiation of the callus begins at the cut end of the explant, and from there it spreads over the entire explant. Now over the period of time, the nutrients that are present in the culture media they are consumed by the cells because they are growing, they are dividing, they are increasing in number, they are increasing in volume. And once the nutrients are consumed from the culture media, the cells stop growing. Once the cells stop growing, we have to replace the media by the fresh media, or we have to remove those calyces from the explant onto the fresh media, which we call it as subculturing. Now, subculturing also helps in the proper maintenance of the growth of the cells. So, once sufficient amount of callus is grown, if you want differentiation of the cells, we can further modulate the composition of the media in terms of growth regulator, and we can induce the differentiation of the different parts, plant parts from those cells, because we discussed that callus is nothing but undifferentiated mass of the cells. So, here you can see there are different stages in callus. First, we take an explant material, which is nothing but a differentiated organ. Once we placed that explant onto the culture media, it enters into a de-differentiation stage, that is meristematic state, where the cells just start dividing without differentiating into the proper organs. So, after de-differentiation, we reach to the callus state. Now, once the callus state is achieved, we can manipulate the hormonal composition in the media, and we can again enter into the differentiated stage wherein we get different organs of the plant. So, this cycles keep on going whenever we are going for subculturing. In various studies, it has been seen that whenever the cells in a calli are exposed to auxins at higher concentration for the initiation of cell division, these cells attain a character of endogenous production of the auxins, and this condition is called as auxin habituation, and those cells become autotrophic for auxin. They themselves start producing auxins, and the same has been seen in case of cytokinins also. It has also been observed that as we enhance the subculturing of a callus, the friability of the resultant calli increases, and once these calli becomes more friable, they become more suitable for suspension cultures. Now, if you want to grow calli just to transfer them in a suspension culture, friability can also be enhanced by modifying the plant growth regulator composition. Like, you can enhance the composition, you can enhance the concentration of auxins in the media, and it has been seen that auxins enhances the enzymatic activity of certain enzymes, which basically causes the dissolution of the middle lamella or the adhering or the cementing material between the two cells, which separate the cells or make the calli more friable, and eventually those can be used for the suspension cultures. Now, if we look into the growth of a callus, there are different phases of the growth. First one is the induction. Induction is nothing wherein a condition wherein the cells of the explant prepare themselves for division, for growth on the artificial media. Once the cells of the explant prepare themselves for proper division, for proper growth, cell division phase gets initiated, and in this phase cells continuously divide onto the media, onto the explant, and the size of the callus grows. This initiation of the cell division is followed by a linear growth period where the cell division rate of the callus, it becomes linear. They keep on continuously dividing till the media gets exhausted. And the last phase is cell differentiation. If you want to enter into the cell differentiation phase, then you have to modify the hormonal composition of the culture media, which will leads to the differentiation of the cells into different organs. So, these are the broadly four growth stages of a callus culture. Now we will discuss about the steps that are involved in the callus culture. So more or less these steps will be the same steps that we have discussed under the steps that are involved to carry out a plant tissue culture experiment. So first one is to sterilize the glass and plasticware that are to be used in the culture media, either by autoclaving or by treatment or by using pre-sterilized glassware or the plasticware. Then you have to select the explant depending upon its age, its size, the quality of the source plant material and so on and so forth, and then you sterile that explant material. You prepare the culture media as per your requirement. Now since we are talking about the callus culture, now callus culture experiments are always done on a solid media. Therefore, one of the important component in the preparation of the nutrient media will be the gelling agent, and as we discussed, there are different gelling agents, agar being the most popular one, followed by agarose or gelin, which is also known as gellerite, and then we sterile the nutrient media which we have prepared. Then we transfer the sterile explant material onto the media, and then we maintain that callus culture by subculturing in the culture rooms or in the BOD incubators. These are the major steps that are involved in the callus culture. Now we move on to the application of callus culture. Now if we look into the steps or the material which we have discussed till now under callus culture, we saw that callus is nothing but undifferentiated mass of the cell, and we know that callus is always grown on a solid media, and this callus can serve as a starting material to initiate single cell or suspension cultures. As we discussed that we can convert those calluses into more friable callus by changing the hormonal composition or by increasing the oxygen concentration, which will make the callus more friable into smaller fragments, which can be used as a starting material for suspension cultures. Or we can subject small fragments of the calluses in the suspension cultures and subject them towards agitation, which will further break down those calluses into smaller micro level fragments, which will help in initiating the suspension cultures. So we can say that callus culture or callus act as a raw material for the initiation of suspension culture. Now callus also provides system for various morphogenetic and physiological studies. We discussed that callus, again I repeat, is an undifferentiated mass of a cell. Now if you want to differentiate that callus into different organs, we manipulate the hormonal composition, we manipulate the composition of our culture media. Once we manipulate the composition of the culture media, the morphogenesis in those undifferentiated cell gets initiated. If we have a composition which favors the shoot development, the shooting of that undifferentiated mass occurs. If we have a composition which favors the rooting of that undifferentiated callus, then the rooting will start. So whatever morphogenetic initiation is there, this will help in studying the exact mechanism of that morphogenesis, and vice versa also. And again, what are the physiological changes occur when a leaf begins to sprout from the callus, or what physiological changes occur inside the cells when a stem part is being initiated or a bud is initiated from the cells. So bottom line is that callus culture helps you in studying the details of the morphogenesis or the physiological changes that are going inside the cell. Now callus cultures can also be used to enhance the production of the secondary metabolites. Now we know that these secondary metabolites act as defense compounds for the plant cells themselves, but these secondary metabolites are of utmost importance because of their potent biological action, and they can be exploited in against various diseases. Therefore, these secondary metabolites are of utmost importance for the pharmaceutical industry. So by manipulating the cultural composition, by manipulating the genetic composition of the callus, we can always enhance the production of these secondary metabolites. And last but not the least, we also discuss about the soma-clonal variation, which occur whenever there is prolonged subculturing from the callus. And these are nothing but, as I told you, that these are the genetic modification or mutation occur in the cells whenever they are subcultured. Because whenever you are subculturing a callus, it enters into a new environment, new chemical environment with respect to the culture media, because every time you prepare a culture media, there will be a slight variation in the composition until and unless you are very much careful in the preparing the culture media. So such kind of not only the chemical variation but the environmental variation also, because you are removing the callus from one place, you are shifting it to a new vessel, onto a new media. Such kind of variation forces the cells to undergo genetic modification, which induces the soma-clonal variation. And many times it has been seen that such variations are very useful, and one can easily identify the useful mutation in the cell, and they can develop a new mutant or the new varieties of such plants. Now we move on to the second type of the plant tissue culture depending upon the maintenance, that is suspension culture. So as the name indicates, suspension, the media in such cultures will be in the form of liquid, and the cells will be suspended in that liquid media. So this is a major difference between a callus culture and the suspension culture. Callus culture is grown on a solid media, whereas suspension culture they utilize liquid media, and the cells are suspended in that media. Now we discussed that calluses can be used as a raw material for initiation of suspension culture, or in simple words, callus can be used as an explant material for suspension culture. But you can also use the explant material that is directly obtained from the source plant, and that explant material can be a part of leaf, can be part of bud, it can be any part of the plant. But as we discussed, that suspension culture is nothing but the suspension of the cells in the liquid media. So we cannot put the entire piece of a stem into the liquid media. We have to first separate the cells of the stem and this separation can be done either by mechanical means or by enzymatic means. Now in mechanical means, you just grind the cells and then suspend them into a suitable media and then filter them or pass them through a suitable sized sieve, which separate the cells, the cells into smaller aggregates, and that can be used in the suspension culture. But the major problem with mechanical method is, during grinding it has been observed that it damages the cells, and this damage decreases the viability of the cells. Therefore, the cells that are obtained by mechanical separation, they have a very low growth rate in the suspension media in comparison to the enzymatic method. In the enzymatic method, we incubate the plant part or the explant material with enzymatic solution for suitable period of time, which is generally 30 to 35 minutes or 45 minutes. Now this enzymatic solution is nothing but it is a mixture of different enzymes like celluloses, hemicelluloses, pectinases or chitinases. These enzymes basically dissolute the adhering agents that is present between the cells, thereby separating the cells. And the major advantage of this method is there is very less damage to the cells, and therefore the growth rate of cells that are obtained by enzymatic separation is higher in comparison to the mechanical method. So once the cells are separated or you achieve smaller aggregates of the cells, you can initiate the suspension culture. Now once the explant material, whether it is obtained from callus or by the source plant material, is subjected or is introduced into the liquid media, agitation has to be provided to that liquid media. Now what is the importance of agitation? Now this agitation prevents the formation of aggregates and clumps of those cells. Now by virtue of nature, the cells have a tendency, or the cell walls of different cells have a tendency to adhere to each other. And if you don't agitate the liquid suspension, then those cells will come close to each other and then get adhere, forming the clumps or the bigger aggregates. So this agitation also not only helps in preventing the formation, but it also helps in breaking the calyces or those aggregates into the smaller fragments or the single cells into the media. This breaking of aggregation or prevention of the formation of aggregates, this helps in maintaining a uniform cellular density into the media. Thereby, consumption of the nutrients from the media is quite uniform in every corner of the culture vessel. And agitation also helps in the aeration of the media, which is important for the growth of the cells. Now, if we look into the composition of the cellular suspension into the liquid media, we can broadly categorize the cellular population into the liquid media into three categories. First is the free or the single cells of different shapes. Now, we discuss that the smaller aggregates are there in suspension culture, and we keep on agitate that culture vessel for proper aeration, for breaking those clumps or to prevent the formation of aggregates. Now, by virtue of this continuous agitation or shaking, we get these three components. From amongst these three, first one is the free or the single cell of various shape. Then second category of cells that are observed in the suspension culture is cell aggregates or the smaller aggregates, which contain up to 10 cells or up to 20 cells aggregate is there, and the group of cells which have thread-like morphology. So, such kind of genetic variation has been observed in the cells in the suspension culture. Now, we move on to the growth pattern that is observed in a suspension culture, and it is more or less similar to that of the callus culture. Broadly, the different growth phases in a suspension culture have been divided into 5 phases, and we get a sigmoid or S-shaped growth curve, as you can see in the picture. And these 5 phases are lag phase, exponential phase, linear growth phase, diminishing growth phase, and the stationary growth phase. Now, the lag phase of a suspension culture is somewhat similar to that of the induction phase in the callus culture. And in this lag phase, the cells prepare themselves because the cells from the explant material or the callus are introduced into the liquid media. The environment, physical environment changes, so they prepare themselves or they acclimatize themselves towards the new environment, and they prepare themselves for the cell division or for their growth. In the exponential phase, the cells start dividing. They start consuming the nutrients from the media and they start dividing. In the linear growth phase, there is a constant rate of cell division, and the number as well as the size of the cells grow continuously. The fourth one is the diminishing growth phase, and since the cells are continuously dividing and growing in size in the linear phase, they very rapidly consume the nutrients of the culture media. And once the concentration of the nutrients in the culture media goes down, the cell division rate and the growth rate of the cell decreases. And here these cells enter into the diminishing growth phase of the sigmoid curve. And the last phase is the stationary phase, where the cells completely stop growing and dividing, and if they are not transferred into the fresh media, they will die out because the nutrients have been exhausted in the media. They will get nothing to grow, they will get no nutrient from the media, they will get no help from the media for their growth, for their division, and eventually they will die out if you do not replace the media with the fresh media. Now, we move on to the different types of suspension culture, and broadly we have batch cultures, we have continuous cultures, we have synchronous cultures. Now the batch cultures are those wherein after initial growth of the cells, we remove the exhausted media, we filter out the cellular material, and then we place that cellular material back into the fresh media. So batch cultures are largely used for initiating single cell cultures. Now cell suspensions that are grown in 100 or 250 ml flask containing 20 to 75% of 20 to 75 ml of culture media, they are continuously propagated by routinely transferring small portions of those suspensions onto the fresh media. We remove a small portion of the suspension culture, we dilute that portion say up to 5 times, then we pass that diluted suspension through a needle of a syringe or through a syringe or through a pipette which is having orifice which allows only small aggregates of 2 to 4 cell or single cell to pass through that orifice, and then we further subculture those smaller aggregates or single cell into the fresh media. The major advantages of batch culture is that it shows all the 5 stages of sigmoid curve, that is induction, the exponential growth, linear growth, diminishing and the stationary phase. We can also manipulate different phases, like we can manipulate lag phase, we can maintain the exponential growth phase in the batch cultures. Now you can always reduce the lag phase during the seb culture by introducing the conditioned media. Now conditioned media is nothing but the media on which the cells were previously growing. So once you introduce the conditioned media during subculturing, the lag phase duration reduces considerably. Similarly, if you want to maintain a continuous exponential growth phase into your media, then you have to go for frequent subculturing with the fresh media. When we say frequent subculturing, it means every 2-3 days we replace the media, we subculture our media, our cells onto a fresh media, into a fresh media. So this is how these are the major advantages of the batch culture. It also has certain disadvantages, like there is a constant change in the cell growth and metabolism pattern due to change in the fresh culture or the media composition, and such kind of constant change in the growth and metabolism pattern and media composition leads to the absence of steady state of the growth, and therefore you cannot study the growth pattern and the metabolic pattern in these batch cultures. So friends, this was about the batch cultures that are used in the suspension culture methodology. In the next lecture, we will discuss about the continuous suspension cultures, synchronous culture and their types. Thank you.